Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. What if the molecules cancer cells produce as part of their metabolism aren't just byproducts, but active drivers of the disease itself? Compounds like lactate, succinate, fumarate can actually reshape the tumor environment and help cancer survive. We often think of metabolism as something passive, just fuel being burned and exhaust being removed. But in cancer, these metabolites can actively change cell behavior, reprogram metabolism, and even suppress the immune system. In this video, we'll explore these so-called oncometabolites and how they contribute to both cancer metabolic reprogramming and immune evasion. So without further ado, let's get into it. So this is going to be the second video about how mitochondrial dysfunction can lead to the creation and the maintenance of cancer in our body. And I left off this last video on this particular graphic here and talking about how these certain TCA intermediates can act as oncometabolites. And that's what this video is going to be about. Let's get back oriented again. So here is a schematic or a cartoon of a eukaryotic cell that we all are. And we're here looking at this organelle called the mitochondria. We're going to zoom in and we're going to make it 2D. And we're going to look at where they have the outer membrane, the inner membrane, the TCA cycle, the mitochondrial DNA, and the electron transport chain, which is where oxidative phosphorylation is actively happening at. And we're going to then kind of show where this is where glucose is coming in through pyruvate. Pyruvate is going through acetyl-CoA. We've looked at these in the metabolism videos that we've looked at earlier in this series. Here's fatty acids, and here's the electron transport chain. So I'm going to zoom in specifically to the TCA cycle for this particular video. So this is in the mitochondrial matrix right here. And so this is another way of looking at the TCA cycle. And we're going to see here that we have, here's acetyl-CoA, here's citrate, here's succinate, here's diisocitrate, alpha-ketoglutarate, et cetera, throughout this process. And these are the substrates that are being converted to the next substrate via an enzyme. Here's an enzyme here, aconitase or citrate synthetase or isocitrate dehydrogenase, et cetera. And then you're going to see a couple of things. You're going to see a green bubble and you're going to see a salmon colored bubble. And the green bubbles are an important reminder of why we all need micronutrients. So here we have iron, magnesium, B1, B2, B3, B5, lipoic acid, manganese, et cetera, et cetera. And these are cofactors for these enzymes to functionally work. So if we don't have those cofactors, then therefore aconitase cannot convert citrate to succinate. Or let's say diisocitrate cannot be converted to alpha-ketoglutarate via this enzyme isocitrate dehydrogenase without B3, magnesium, and manganese. And then on the flip side, in the salmon color bubbles, these are toxins that can inhibit these enzymes from actually working. So here's fluoride, here's mercury, arsenic, and then a bunch of different types of either reactive oxygen or nitrogen species. And then that's going to make aconitase, even if it has the cofactors working there, it's not going to be as effective or efficient. Why do I care, about this at the end of the day? Because when we have a functional deficiency in either CoQ10 or magnesium or B2 in this particular incident, then succinate cannot be converted effectively to fumarate as well. So now succinate is going to build up, or fumarate, because it can't be converted to malate, is going to build up. And that is where the problems start to begin. So I'm going to now pull this paper here that was published in January of 2020, and it's titled Mitochondrial TCA Cycle Metabolites Control Physiology and Disease. And it says here, mitochondria are signaling organelles that regulate a wide variety of cellular functions and can dictate cell fate. Multiple mechanisms contribute to communicate mitochondrial fitness to the rest of the cell. Recent evidence confers a new role for TCA cycle intermediates, generally thought to be important for biosynthetic purposes as signaling molecules with functions controlling chromatin modifications, DNA methylation, and hypoxic response, and immunity. This review summarizes the mechanisms by which the abundance of different TCA cycle intermediates control cellular function and fate in different contexts. We will focus on how these metabolites mediated signaling can affect physiology and disease. And I like this paper because it actually has really nice graphics. So here is some high level of what the mitochondria can do to communicate that there are issues within the cell that need to be addressed. So if the mitochondria was to lyse itself and this wall was to be damaged, and cytochrome C which is part of the electron transport chain, cytochrome C is in the electron transport chain. If it was to leak out, that would lead to caspase activation and apoptosis through the intrinsic pathway. If there are buildups of hydrogen peroxide or other oxidative markers, that can change gene expression or AMPK or mitochondrial DNA, etc. What we're going to focus on in this particular video is about these TCA intermediates and other oncometabolites and how they can change gene expression and or can stabilize certain intermediates that can lead to metabolic reprogramming within cancer. This is critical for us to understand the Warburg effect in a future video. So this is another picture of where these intermediates are coming. Here's acetyl-CoA, here's itaconate, here is alpha-ketoglutarate, here's L2HG, here's succinate, etc. And they're looking at different ways that it can either modulate the immune system or stem cell function or our body's heat mechanisms. But I am in particular really interested in tumor genesis and immune modulation because that's going to lead us to cancer creation, maintenance, and that cancer's escape from the immune system and being recognized. Here's another picture from the same paper looking at how different TCA intermediates can then interact with the genome in the nucleus and actually change gene expression. And I'm going to go to a paper that was more recently published in September of 2025, just a few short months ago. And it's titled TCA cycle derived oncometabolites in cancer and the immune microenvironment. Oncometabolites are aberrant metabolic byproducts that arise from mutations and enzymes of the TCA cycle or related metabolic pathways and play central roles in tumor progression and immune evasion. I will correct this paper. It does not have to be a mutation in an enzyme for this to happen. You can have a functional deficiency or toxicity that will lead to this. And then once you have any degree of mitochondrial and or cytosol extracellular phosphorylation, which is a phenotype seen in all cancer, that by itself can do it. There are no mutations in these enzymes that are required. Just as a side note. Among these, 2-hydroxyglutarate, 2-HG, succinate, and fumarate are the most well characterized, acting as competitive inhibitors of alpha-ketoglutarate dependent dioxygenases to alter DNA and histone methylation, cellular differentiation, and hypoxia signaling. More recently, an immunometabolite predominantly produced by the activated macrophages has been recognized for its dual roles in modulating inflammation and tumor immunity. These metabolites influence cancer development through multiple mechanisms, including epigenetic reprogramming, redox imbalance, and post-translational protein modifications. Importantly, their effects are not limited to cancer cells, but extend to various components of tumor microenvironment, such as T cells, macrophages, dendritic cells, and endothelial cells, reshaping immune responses and contributing to immune suppression. In this review, we highlight the emerging insights into roles of TCA cycle associated oncometabolites in cancer biology and immune regulation. We discuss how these metabolites impact both tumor intrinsic processes and intracellular signaling within the tumor microenvironment. Finally, we examine therapeutic strategies targeting oncometabolite pathways, including mutant IDH inhibitors, alpha-ketoglutarate mimetics, and immunometabolic interventions with the goal of restoring immune surveillance and improving cancer treatment outcomes. I like this paper because it has a lot of great graphics. So here's some of the classic oncometabolites, 2-HG, fumarate, and succinate. You can see how multifaceted these things can be to lead to a cancer type problem. So here is epigenetic regulation, basically changing gene expression. Here's redox balance and homeostasis, aberrant metabolism. This is the reprogramming of metabolism. And then pseudohypoxia, what I mostly focus on, which is related to aberrant metabolism, and then genomic instability and post-translational modifications, which are also important. Here's another slide of looking at how the same metabolites, itaconate, fumarate, succinate, and we'll look at others in the next couple of slides, as being able to modulate the immune system and change the immune system from being able to detect cancer to being not able to detect cancer, or in an immunosuppressive state that leads us not to have the same effect from our innate immune system or our natural immune system, or even having effects on immunotherapies as well. Next, I'm going to zoom in on something called pseudohypoxia. And pseudohypoxia is basically where if the cell is not hypoxic, or there's actually normal levels of oxygen, then there are certain conditions that can lead to the stabilization of the hypoxic response element through a protein called HIF1 and HIF2. HIF1 stands for hypoxia-inducible factors. These are factors that are very important for the metabolic reprogramming and immune evasion that happen within cancer. So if we are stabilizing these hypoxia-inducible factors without having oxygen, we call that pseudohypoxia, because it's basically the same response that you would have if you had low oxygen, but there's no low oxygen that's leading to this problem. This is personally where I believe prevention is huge. Pseudohypoxia can be triggered by many things. We'll actually talk about that. And I do think that if those same environmental factors are in play while you have cancer, it's going to continue to lead to continual stabilization and disease progression. And so removing these pseudohypoxic factors are critical. Let's talk about some of the major pseudohypoxic factors. So one of the major pseudohypoxic factors is called lactate. Lactate is a major oncometabolite. Lactic acid will stabilize hypoxia-inducible factors and lead to metabolic reprogramming, but it doesn't just do that. Lactate will also be transported outside of the cancer cell and lead to something called the acidification of the tumor microenvironment. And that acidification of the tumor microenvironment will then tell the immune system to back off, will lead to metastatic spread, and will lead to chemotherapy and likely immunotherapy resistance as well. So this oncometabolite not only protects the tumor from immune cell response, but it also will lead to further metabolic reprogramming and continuation of the vicious snowball that is created with cancer's aberrant metabolism. And then lactate can also be responsible for, if that wasn't enough, lactate can also be responsible for the cancer-related cachexia that we see, as one of the mechanisms is that lactate will actually change our metabolism to where we will expend extra energy. So our caloric needs will go up because of lactate. And if we don't meet those caloric needs by intake of calories to maintain our body weight, we'll end up losing weight. And as most people who have cancer know, they get a low appetite. They are on treatments that will cause nausea, vomiting, and upset stomach. So therefore, it becomes kind of another vicious cycle where the lactate is leading to an increase in metabolism. And then because you have decreased oral intake because of nausea, because you're just not feeling like eating, that's going to make even a worse caloric deficit, and then you're going to have rapid weight loss. And then succinate, the other major oncometabolite, is also at play here. It can lead to both stabilization of hypoxia-inducible factors and metabolic reprogramming, but it can also have effects on cell migration, making new blood vessels, which can lead to cancer growth and metastases. And so it brings me back to one of my favorite graphics that I've ever seen by Seyfried's group. It's this graphic right here. And we'll see here is we'll see glucose and we'll see glutamine at the top. And we'll see at the bottom is we'll see lactic acid and we'll see succinic acid. So when glucose is fermented, it will make lactic acid. And when glutamine is fermented, it will make succinic acid. So that lactic acid and succinic acid will stabilize HIF, like it says here, stabilizing HIF. And that will lead to further metabolic reprogramming. But the other thing that lactate and succinate do, besides acting as an oncometabolite in that particular way, is that it also acts as an oncometabolite by this extracellular acidification. And that's what leads to failure of the immune system to recognize the cancer and immune escape. And so therefore, when we look at a very high level overview of what Seyfried proposed in 2017 with this graphic right here of putting people on a ketogenic diet and inhibiting glucose and inhibiting glutamine, it would make sense then by limiting the amount of glucose getting to the cancer cells by being on a ketogenic diet, or by adding a glucose inhibitor or a glutamine inhibitor, we're going to be able to partially or hopefully completely shut down glucose and glutamine being fermented into lactate and succinate. Then you're going to see that makes a whole lot of sense from a variety of ways, not only for the vicious snowball that's happening from the stabilization of hypoxia-inducible factors and the metabolic reprogramming that happens because of that, but also because when we shut this down of lactate and succinate, now the extracellular environment is less, as you would imagine, acidified. And now conventional therapies such as chemo and immunotherapies work better, and our own immune system works better at recognizing the cancer. There's less immune escape. There's more of a chance for us to have a significant effect at hopefully killing and eradicating the tumor. So that's why this protocol as a gestalt, which we're going to get into in future videos in greater detail, why this makes so much sense on so many levels. If you like videos like this, please like, share, and subscribe. And until next time.